A Carnegie Institution for Science audio press release for October 28, 2014. Right place, right time. Cellular transportation compartments. Proteins are the machinery that accomplishes almost every task in every cell in every living organism. The instructions for how to build each protein are written into a cell's DNA. But once the proteins are constructed, they must be shipped off to the proper place to perform their jobs. New work from a team of scientists led by Carnegie's Munivar Aksoy and Arthur Grossman describes a potentially new pathway for targeting newly manufactured proteins to the correct location. Their work is published by the journal The Plant Cell. The team's discovery concerns a cellular organelle that has been called an acetylchalcosome. It is a compartment that isolates potential harmful or disruptive compounds from the rest of the cell and is also involved in the turnover of cellular components, similar to the so-called lysosome in animals. They're rich in phosphate-containing molecules, and the team noted that they build up to high levels when cells of the single-cell green alga Chlamydomonas are deprived of sulfur. They discovered that acetylchalcosomes are also surprisingly involved in targeting proteins out into the cell space between the cell's membrane and the cell wall. Working with Chlamydomonas, the team, which also included Carnegie's Warulda Putakam, was examining the organism's responses to nutrient deficiency. They found that mutant cells, lacking the ability to form these acetylchalcosomes, also lack the ability to cope with sulfur and nitrogen deprivation adequately. What appears to happen with these mutants is that the proteins that specialize in helping the cell survive a deficiency of sulfur or nitrogen don't get shipped out to the space between the membranes and cell wall where they're needed. Because of this, feedback is sent to stop construction of the proteins and the messenger RNA that encodes those proteins, and the entire response to nutrient deficiency is derailed. Grossman said that their findings point to a novel way to target proteins to where they're needed to function, and indicate that this targeting is also associated with checkpoints that might control gene transcription. These findings tie in with new work from Sabiwa Merchant at UCLA, with whom Grossman's lab will be working to explore the interrelatedness of their discoveries. Merchant's team used a combination of various spectroscopic methods to visualize a metal-storing compartment in Chlamydomonas that may be similar or related to that studied by Grossman. The compartment studied by Merchant specializes in storing copper and in making it available when it's necessary for the construction of certain proteins. Their work is published in the journal Nature Chemical Biology. Merchant said the researchers wonder whether there are actually different types of acetylchalcosomes or vacuoles in any one cell, each housing different amounts of individual metals. The scientists look forward to working together to explore the new possibilities in understanding mineral-nutrient metabolism brought to light by their two papers, which underscores the interplay between multiple pathways for maintaining normal cellular function. This image is of acetylchalcosomes in a sulfur-depleted Chlamydomonas cell, courtesy of Munivar Aksoy. The polyphosphate in acetylchalcosomes is easily seen. This image is of a sulfur-replete Chlamydomonas cell. And this is an image of a Chlamydomonas cell with polyphosphate in the walls, where it's been directed. With the independence for nimble pursuit, Carnegie scientists tackle some of the most profound challenges in modern science, fundamentally changing what's possible for us, our planet, and our universe.